So, good morning all. I welcome everyone to this Gyan course on the fourth day on the Gyan course on biofluid mechanics, mathematical modeling and challenges. So today Professor Yun will be covering a topic on nanoparticle transport in solid tumors. The contents include multiphysics model of drug delivery, radiotherapy enhancement, thermosensitive carriers, and then she will cover some case studies, and then after that we'll have some question and answer session. After that we'll discuss. I request now Professor Yun to continue her lectures.
Good morning, everyone. So we're going to look at new topics today, and that is the multi-scale multiphysics modeling of drug delivery to solid tumor. So this is the outline. We'll first look at the problem description. So obviously when we talk about drug transport for the treatment of cancer, the problem is different from blood flow in the cardiovascular system that we discussed in the previous lectures. And we're going to look at the basic mathematical models. I'm not going to show you the most advanced model in this course. And I hope that you can make some connection with the basic fluid mechanics and the mass transfer equations that we covered in the previous lectures. You will see some of the equations familiar in this case. And then I'll show you the modeling strategy to try to simulate thermosensitive liposome-mediated drug delivery for the treatment of cancer. And then I'll show you another example that involves the use of nanoparticles to enhance radiotherapy. Even though we're not going to model the radiotherapy itself, but we're going to look at the nanoparticle distributions in solid tumor. And these are the special nanoparticles designed to enhance the efficacy of radiotherapy. I will touch on a little bit on system-based models. So that will probably involve a little bit of system biology of drug transport and cellular uptake. Okay, so looking at drug delivery in solid tumors, so the first we need to know what the problem that we're facing and looking at the tumor vasculature and also the tumor microenvironment. So here we have two figures. The one on the left shows a normal vasculature. And well, reason we're interested in the vasculature is because the chemotherapy drugs are usually delivered through intravenous infusion. So they come through the systemic circulation. And so they need to be carried in the blood and then to reach the tumor site. And there that get transported, diffused and convected through the capillary vessels into the tumor cells. So we can see the differences between a normal vasculature and tumor vasculature. So their morphology is different. Tumor vessels are more tortuous, curved, and the tumor vessels are also more leaky. So the vessel transport properties are different. And this is the tumor microenvironment. And if you look at the vessel, this is a microvessel and the surrounding environment where the tumor cells see. So you have a comparison of the normal tissues and the tumor tissues. Normal tissue on the left, tumor tissue on the right, and again, and you can see the more complex tortuous vessel structures, and some differences in the extravascular space where the tumor cells reside. So now thinking about drug transport in solid tumors. So as I said earlier, that the anti-cancer drug will come into the system through systemic circulation through the blood flow. So we first need to understand how the blood can be transported through the circulation. And then we need to look at the tissues around the microvasculature, so the figure in the middle, where these are the tumor cells, and we need to look at how the drugs that is carried within the blood through the blood vessels, how they can transport across the vessel cells into the tissue and then to reach tumor cell and then to exert the effect. So when they reach the tumor cells, it obviously it depends on, there are different ways to model that, and the most accurate way to model the effect of drug on tumor cells is to understand through the cellular signaling pathways. So this is the problem description. And the strategy in terms of developing a mathematical model to simulate this entire process is obviously very challenging. So the first part is we need to understand blood flow in tumor vasculature, and then that is connected to drug transport through the tumor extravascular space. And then once the drug reaches the tumor cells, there is a drug uptake and effect on tumor cells, so whether it's sufficient to kill the cells or not. So in each of these processes, and the first two, it will depend on the tumor physiology, and that includes the complex vasculature, and it will involve blood flow, which can be abnormal, impaired blood flow in tumor vasculature, and also very difficult interstitial transport. Now, when we look at the drug uptake and effect on tumor cells, and that would involve cellular response, and that typically involve complex cellular signaling transductions and interactions, and also depends on the mode of drug delivery. So clearly, this is a multi-scale, involves multi-scale complexity and challenges. So there have been several review articles in this space. Listed over here is one of our own review articles written with the PhD students, but you can find a lot more in the literature. There is a fairly rich body of literature in this space. So how to model all of these? Well, when you have complex problems, you might have to break it down sometimes and just to tackle one aspect at a time. So the first part is to look at the pharmacokinetics and pharmacodynamics based models, and these are usually compartmental models. And but these compartmental models' advantage is that they usually involve a system of ordinary differential equations. Mathematically, they can be solved relatively easily and quickly, but they only give you a temporal distribution, so they don't, because you don't resolve the transport in space. So you only have the time variation of the variables you're interested in. And then looking at there's microscopic transport models, and these typically involve a tumor cord model, so that's kind of similar to the Croft cylinder model, where you have a tumor tissue surrounding a single capillary vessel that feeds into the tumor. That's called the tumor cord model. And there are also tumor model with embedded vascular networks. And then looking at the macroscopic level, we have macroscopic transport models which can be solved in either 2D or 3 dimensions, and these give you a spatial temporal distribution. So if you solve a time-dependent problem, you can get a distribution, spatial distribution of the drug concentration, and then you also have a temporal distribution. You can average it over space and look at the variations over time. Now, so these are about the transport models for the delivery of normal drug, like in the free form. And one of those common drugs is called doxorubicin, DOX. And because the standard chemotherapy drugs has a serious side effect, so over the past maybe more than 20 years, 30 years, that material scientists and nanotechnologists have developed nanoparticles, and specifically for targeted delivery and controlled release of chemotherapy drugs. And one of those special nanoparticles developed, there are whole different type of nanoparticles. I'm not going to cover all of those. I'm just going to talk about one specific type that was included in our model, which is a thermosensitive liposome. So thermosensitive liposome, I'll come to that later in the slide to explain what it's about. So we developed a multiphysics model to simulate the delivery of thermosensitive liposome drug delivery system to solid tumor. Now there is another branch of models which involves molecular dynamics modeling of nanoparticle drug carriers. I'm not going to touch on that. And there is a system-based model of drug transport and cellular uptake. I'm going to touch on that just a little bit, so you have a little flavor of what it's about. And obviously, multi-scale model is important. You have the fluid and drug transport different length scale. And also there are multi-scale models specifically to deal with the kinetics at different scales. So that's the kinetics different time scale, for example. Right, so let's start with a compartmental model. So this is a PK/PD-based compartmental model for thermosensitive liposome drug delivery. Now so this model can actually be reduced to the standard PK/PD-based compartmental models for a standard chemotherapy drug. All you need to do is to remove all the components that's related to thermosensitive liposomes, and then you will come with a basic model for the delivery of a standard drug. So the model consists of, on the higher level, it has three major compartments. So we have systemic plasma. This is one of the compartments, and then you have the tumor compartment. And the tumor compartment actually consists of three subcompartments. So you have the tumor plasma, tumor extravascular, extracellular space, and also the tumor intracellular space. And then we have another big, the lumped tissue compartment. So you work out the connections between each of these compartments. And for example, you start with a chemotherapy drug. So there is a dose injected. So here are a few symbols because we are looking at building a compartmental model for thermosensitive liposome drug delivery. So the liposome drugs are these symbols. So they're injected into the system through intravenous injection, and so they can come in, and then they can also get eliminated by the body through the system. And then so they can release part of the drug. So this is the chemotherapy drug. So the drug it comes into the system in encapsulated form. It can be released through the trigger, and here the trigger is temperature. So part of it can be released even at the body temperature with a small leakage. And then there is communication with the tumor compartment through tumor plasma, so through the blood transport into the vasculature in the tumor space. And over here, if the temperature is right, then controlled release takes place, and then the drug is released from its carrier, and so that you have a different release rate, which could be much higher than the body temperature. And then you have other internal communications through transvascular. So these components relate to the transvascular transport into the tumor extravascular, extracellular space. And then there is also through the cell membranes to enter into the tumor cell space. And there are obviously communications between the systemic plasma and tissue as well. So you can formulate all of these and write the relevant equations. So the aim of using this kind of model is to try and to have some sort of understanding about the interplay among these different transport steps, and also to identify which parameters, because we were going to encounter a large number of parameters. So to understand which parameters have the greatest influence on the peak intracellular drug concentration. So the key parameter we are interested in here is the drug concentration within the tumor cells. So we call it peak intracellular drug concentration. The higher of this intracellular drug concentration, the more effective the treatment could be. So these are some of the equations. The equation is actually much long and then long list, and I don't intend to include all of these, but I've cited, I think the paper will come later. In the paper you can get hold of the whole list of the equations. So we do need to write down the list of the ordinary differential equations. You start with a different compartment, and for each compartment, for example, you start with the PK model of the thermosensitive liposomes in the systemic plasma, and then assuming you have a bolus injection, and you have that equation, and then you can also do the pharmacokinetics of free drug. So once it's released from the nanoparticle carrier, and then it's transported in the systemic plasma. And likewise, the equations for describing the transport free drug in tissue compartment. And then you can also write the system equations for the TSL and also free drug in the tumor plasma, and so on. So you write each of these equations for each of these compartments. And then this equation can be connected and solved mathematically. So here is just a map of the properties, the parameters that you need to fit into the model. So I've clustered them into different categories, and you can look at that in terms of the drug properties. That we have these pharmacokinetics and the release kinetics. These are related to drug properties, and there are other parameters that's related to the drug, including the clearance rate of the liposomes, clearance of the free drug, body temperature, and in this case, because we're talking about thermosensitive liposomes, the release of the drug is triggered by local heating, so it requires a mild hypothermia. So hypothermia temperature is also necessary. And when we look at the tumor physiology includes these various parameters like the vascular permeability, and typically in tumor that the vascular permeability is much higher than in normal vasculature. And then the vessel density, because in this case one of the practical problems is that you may not model the individual capillaries or individual vessels within a tumor. So you might want to use a volume average approach. So somehow you need to estimate the vessel density distribution within a tumor. And the blood perfusion rate, obviously that's going to influence the transport and then the tumor cell uptake. And then in terms of the treatment, which is specific to TSL-mediated drug delivery, this including the heating algorithms. So how much heating is applied. So heating duration, timing, so whether you heat it continuously or you apply post-heating, so you heat it for a while, you stop and then heat again. So there's a different type of protocols for heating. And also the feasibility of precise heating, of course. And there is also a question about whether the release is going to be triggered within the vasculature, so it's intravascular triggered release, or outside of the vasculature, which is extravascular triggered release. So there's lots of these considerations need to be made, and also lots of parameters to be considered when you do such a model. So it is interesting and important to actually do a relatively simple sensitivity analysis, and just to have sort of a qualitative understanding about how each of these parameters might influence, and which one is less important, and which one is likely to be more important. So this was the work that I did with my PhD, well, the PhD students. At the time when she did this work, she already finished her PhD, so she was working on the postdoc project. So just formulating, solving a system of ordinary differential equations we described earlier using this multi-compartmental model, that she did a sensitivity analysis just by modifying each of these model parameters. I know it's difficult to follow the symbols, all of these. It doesn't really matter, just to trying to get some feel about how changing each of these parameters is going to affect the normalized intracellular drug concentration, because here the targeting parameter is to trying to maximize the intracellular drug concentration. So for instance, what this bar means is that it means increasing this parameter by a certain amount, that it has a negative, so it reduces the intracellular drug concentration by this amount. So when you look at this kind of map, this is done for continuous heating or pulse heating for 1 hour. But basically, the trend is actually similar. So the key players are still, for instance, here, we have the KR42, which is the drug release rate from the thermosensitive liposomes at mild hypothermia temperature, 42 degrees centigrade. So this is seems to be a key player, and it's still the tolerant over here. And what this means is that if you increase these by a certain percentage, then it has a quite dramatic effect on increasing the peak intracellular drug concentration. Whereas if you look at this parameter, this is related to the blood perfusion rate. And if you increase the blood perfusion rate, it has a negative effect on the intracellular drug concentration. And likewise, if this parameter is also can be noticed, PS, this is a product of vascular permeability and surface area within the tumor. So, and then this also has a quite pronounced effect. So based on this kind of analysis, you're trying to identify the parameters that are likely to have the greatest influence on the outcome you're interested in. And then you can study those parameters in a bit more detail. So for instance, we've picked up a few of these parameters. One of those that I mentioned, I think this one is effect of drug release rate from thermosensitive liposomes. Right. The drug release rate, well, this is something, obviously from a modeling point of view, and you can't just pick any release rate, any random number. It has to be realistic. And this depends on the specific nanoparticle design. So the way we did was first to look at the literature reporting this particular type of thermosensitive liposomes and see the range of that parameter, the release rate reported by different research groups, and try to identify the range and then set a baseline value to start off with. And obviously, as the technology advanced for the making of these type of nanoparticles, then researchers, different groups have trying to increase the release rate at mild hypothermia temperatures. So all the range that we experimented with are within a realistic range that can be achieved. So when you do this kind of sensitivity analysis, you can't just consider it from a mathematical point of view. Mathematically, you can increase or decrease the parameter in any way you like, but in reality, that might not be realistic. So what we're looking at here, we have a few curves. Each correspond to, so these are normalized. And so if you regard the green curve as a baseline, so that has a ratio of 1. And then the blue line, and that corresponds to the intracellular drug concentration as a function of time when these release rate was reduced by half. Then if the release rate was increased by 10%, by tenfold, then you get these red curves. So you can see that obviously increasing the release rate from the nanoparticle actually had a dramatic impact on increasing the intracellular drug concentration. So then you can look at the figure on the right. It shows, so we've got two curves there. Now these two curves correspond to, one corresponds to persistent heating, continuous heating, whereas the red curve corresponds to 1 hour post heating. So the continuous heating gives you a higher intracellular drug concentration, which is easy to understand because the release rate is temperature dependent. And if you heat the tumor continuously, you're more likely to sustain the level of high temperature required to achieve the higher release rate. But then the interesting observation is that, so this curve is plotted, the one on the right is the peak intracellular drug concentration as a function of these normalized release rate. And you can see that initially, as you increase the release rate by something up to maybe around tenfold, maybe even less than tenfold, that you have more or less linear increase in the peak intracellular concentration. But after that, the rate of increase slowed down, and then it plateaued. So what that tells us is that, and perhaps further increasing, because there is interactions with other transport mechanisms, and increasing this release rate further would not help increasing the intracellular drug concentration. So, and potentially this could be fed back to the experimentalists and to say that even if they can optimize the nanoparticles, but there's probably no point of increasing it even further beyond a certain point. So it helps with some fundamental level of understanding about how this parameter behaves in connection with the other transport mechanisms. And then if we pick another one, like the blood perfusion rate, which also from the sensitivity analysis, it shows a relative profound influence on peak intracellular concentration. And here, if we again plotted these and looked at, so predicted random simulation for different blood perfusion rate under persistent heating and post-heating. And again, so always that with persistent heating, that you get higher peak intracellular drug concentration. And we can find that the influence of this parameter is actually not linear. And when the perfusion rate was very low, then increasing the perfusion rate increased the intracellular concentration. That's easy to understand because it enhances the transport. Whereas when it reached a certain point, that increasing the perfusion rate actually caused the intracellular drug concentration to drop. And that's because the transport is two ways. You can, so the convection, it can go in and out. So if the perfusion rate is too high, then it means that some of the drug can actually exit the tumor exit the tumor tissues and then leave the system entering into the vasculature. So that's the transport, and it's driven by the concentration gradient. So if the concentration inside the tumor tissue is too high, then it drives the transport backwards, right? And if you look at the now the figure on the right is about the effect of heating duration. So with persistent heating, you can heat it for 1 hour, you can heat it for longer period of times. Does it matter? And it looks like it does. So heating it up to 2 hours seems to be optimal, and heating it to further more than 2 hours, it doesn't make much difference anymore. So this also gives us some sort of guidance about, if this particular type of nanoparticle drug carrier, should it be adopted in the actual treatment, then this kind of models could help understand or design the treatment protocol. Right, so we talked about these compartmental models, and it gives you some sort of qualitative understanding, and then it gives you the temporal sort of variation of the parameters of interest, but it doesn't give you a spatial distribution. So as we have observed with the blood flow, we always wanted to see how it's spatially distributed, and so that it will bring sort of visualization a bit closer to reality. So here, imagine that we have this idealized model. So here we have this region, and we have the blue area represent normal tissue, and then we have this yellow area, and assuming this is a tumor tissue. So this is a tumor within a normal tissue. And then, so here in the tissue, we have blood vessels, and the red ones are the blood vessels, and that's where the blood comes in, and also the drug enters into the normal tissue and the tumor tissues through the blood vessels. And we also have lymphatic vessels, and especially in the normal tissue, so this helps drain the fluid away from the tissue. And usually, for tumor tissues, it has been found that there is a dysfunctional lymphatic system, so that you don't usually model the lymphatic system within tumor tissue. Or if you do include the lymphatic system, then the drainage should be reduced significantly compared to the drainage effect in the normal tissue. So then you can use the standard transport equations that we described before. For example, if the scale of the tumor that you're interested in is relatively large, then the Darcy's law might apply. But if you want it to be safe, you can apply Brinkman equation. So that's all fine. And then you have the mass balance. But here, when you write the mass balance equations, you must consider the source term and the sink term. So the source term is typically the flow through the blood vessels that perfuses the tissue. And then the sink term, in this case, will be the lymphatic vessels, the drainage through lymphatic vessels. And then you couple this with the drug transport equation, this equation that we've seen it in the first lecture, standard equation describing, but obviously you need to apply these to all the species that you're going to model. So for instance, if you model nanoparticle as drug carriers, you need to solve these for nanoparticle transport. And then as the drug is released through the nanoparticles, the free drug also transports in the system, and then you have to solve a separate equation for each of the species of interest. And finally, you get a concentration distribution. And then what it does is that, well, you have the concentration distribution within the cells, and then you wanted to know how what is the response of the cancer cells to that, so the effect of the drug on cancer cells. And this is typically using a pharmacodynamics model, so the PD model, which depends on, but this obviously the exact PD model would depend on the drug and also the type of the cancer cells that you're modeling. But generally speaking, it involves the equation that looks like this. So you get, this is the cancer cell density. So after the treatment, the cancer cell density is expected to decrease, right? So you start off with a given cancer cell density, and after treatment, some cancer cells are killed and the cell death takes place, and therefore the cancer cell density is expected to decrease. So all of these parameters can then be solved, and then you can look at monitor the effect of the treatment. Right, so if we apply these to model the drug transport, thermosensitive liposome mediated drug transport. So we got the compartmental model that we're already familiar with, and we also need to connect the compartmental model with the drug transport process. So the schematic diagram is shown here that the drug comes in through the blood vessels that are within the tumor. So this is within the tumor. So you have lots of these blood vessels, and they carry the drug inside. And then the drug will transport within the vessel, but also the vessels are quite leaky in tumor, so that the drugs can leak out of the vessels. And there is a transvascular transport, and they enter into the tumor space, tissue space, transported by the interstitial fluid. And in this space, and they can stay outside of the cells. They can also enter the cells. So that depends on the transport ability of the drugs, and also depends on the resistance and the cross membrane transport. So coupling all of this together, the aim is to use the model trying to, for any treatment, is to maximize drug accumulation within tumor. So you want as much drug accumulated within the tumor as possible, and you want to minimize the drug concentration in systemic plasma. And the reason for that is very similar to the thrombolysis treatment, is that most of the chemotherapy drugs are they're highly toxic. So if you have a high concentration of chemotherapy drug in systemic plasma, then it can cause other disease. So even though you probably have treated the tumor, but you might have caused other disease, for example, heart disease. And some of the chemotherapy drugs are known to they can cause harm to cardiac tissues or cells, for example. Okay, so just give you a little bit of flavor about how this kind of, what you get from these kind of models. So your starting point is very similar to when we model the blood flow in the vasculature, that sometimes you can get hold of the MR scans of a patient who has a tumor. So this is actually one slice of the MR scan to show. So this is the normal tissue area that we're interested in. The tumor is highlighted in red. And so this can be segmented from MR scans. So likewise, you can build a realistic shape of the tumor. If you look at the 2D geometry, it looks like this, and it's surrounded by normal tissue. And then when you build a 2D or 3D model, you can't just isolate the tumor. You usually have to include the surrounding normal tissue to a certain extent, and so that it allows you to prescribe some sort of boundary condition at the interface between the tumor and the normal tissue. And then you use the model to compare different treatments, administration modes. For example, for a given drug, it can be administered through bolus injection or continuous infusion over different durations. And you can use different dose levels. So this obviously dose levels are within the approved, regulatory approved dose levels. And then within that dose level, you can play with the different injection protocols and to see whether the injection protocol actually have an impact on the drug accumulation within tumor, because that's something we're trying to optimize and maximize. And in terms of mission schedules, administration schedules, you can have a single or multiple administrations within, for example, 2 days. Right, so we can have the 2D or 3D model allows you to look at the spatial distributions, but you can also look at the temporal variations, whereby you average the concentration within the tumor volume, and then look at how it varies as a function of time. So here, I think the examples shows, so on the left that you have a set of curves, and these are the doxorubicin intracellular concentration within the tumor as a function of time. So you can see that from 0 to 9 hours, and these correspond to different injection mode. You have a bolus injection, the blue one. So with bolus injection, you introduce the drug in the bolus. So not surprisingly, you have a peak intracellular drug concentration earlier on. And then as you increase so for the same dosage, the same amount of drug, but if you infuse it slowly over an hour or 2 hours or 3 hours, you get different, you get a spread of these over a period of times. And obviously with the bolus injection, that after 5 hours, the intracellular drug concentration dropped to 0, and that's because part of the drug has been taken up by the cells and the others part has been eliminated through the body. So if you wanted to measure the efficacy of the treatment on the cancer cells, so one of the parameters we look at is the fraction of survival cells. So obviously you started off with 100%. So if none of the cancer cells is killed, then you have 100%. And then ideally, you want to kill all the tumor cells, but that doesn't happen. And so you can compare this different injection mode. With a bolus injection, even though you get a really high peak earlier on, but the effect does not last for very long, and so that the final tumor cell survival fraction, it gets plateaued around this point. Whereas with the continuous infusion over a longer period of times, you have a more sustained tumor cell killing, and so it can reach a lower level of survival. So the lower, the better. So what these simulations actually tell us is that the continuous infusion is more effective at cell killing than a bolus injection. And over here we look at the dosage effect, where the dosage effect is fairly intuitive, so that the higher the dose, so you chose your fix the injection mode, and then you only change the dose. So you have a higher dose. So not surprisingly that the highest dose gives you the highest peak intracellular concentration, and you can see how varying the dose within the realistic range could impact the tumor. The same measure which we use is the fraction of survival cells. So that's how this dosage variation could impact on that. And there are other parameters that you can play with. For example, the single infusion or multiple infusion. So we modeled these using this different color curve to correspond to single. So this one has a high peak at the start and then it tails off. Twice injection, so you have 2 peaks. And then 4 times infusion, you have 4 peaks. And 8 times infusion, you got 8 peaks over this period of times. And then how is that reflected in terms of the treatment effect? Measured through the survival fraction, and you can see that the multiple injection obviously give you a better performance, but the gap between these seem to start to narrow. So that's something quite interesting that you can play with through the model. And one of these realistic applications, and this was done by one of my former PhD students, and we're trying to model a liver tumor, but obviously the model is very, when we say liver tumor, it just means that the particular tumor model was built based on a patient who had a liver tumor. It does not necessarily mean that the tumor characteristics reflect liver tumor, because all the models that I show you today are actually very basic. It does not consider tumor-specific properties, because those parameters are very difficult to get. So just from the transport point of view, that we build a this liver. This is the normal tissue extracted from MR scans of this patient, and then this is where the tumor sits. So tumor is close to the edge of this tissue that we reconstructed. Well, I think from the modern point of view, the issue that we wanted to address here is that in the standard basic models, that you assume the tumor tissue has a uniform homogeneous structure properties throughout the tumor tissue, but that is not realistic. In reality, that tumor tissue actually has very heterogeneous properties. But then how can that be modeled? It's actually very difficult to do it. And then we had the opportunity to do it to a certain extent in this case, because the radiologist who obtained MR images for us, he had a special MR protocol, which allowed him to look at the perfusion through the tissues, through the tumor, and then which kind of correlate with the permeability through the tumor tissue. So it's not a direct measure, but it's relative. So based on that information, and you can see that we divided the tumor into multiple regions. I think we managed to do it up to 6 regions. And then each region we have calibrated, because this permeation through the tissue somehow correlate with this parameter in our model. This is the surface area to volume ratio, which reflect the vasculature, how well vascularized the tissue is. And then this parameter obviously has an impact on the transport. So we're able to get this number, and then this was scaled based on the MR signals that we obtained. So at least this allows us to identify or differentiate the region. So each of these regions in this order that has a higher S to V ratio, this means this region is more vascularized than this region. So then that allows us to look at this intracellular drug concentration, which is the parameter that we're interested in, as a function of time in different regions. So you have the well-vascularized region, not surprisingly, that it has the highest intracellular drug concentration, whereas the least vascularized regions has the lowest peak intracellular drug concentration. And then you can look at the other measure as well, the survival fraction of tumor cells. And so again, that corresponds to this trend. Well, interestingly, it's not. I think it's actually it's not quite exactly the same. So this is a well-vascularized region, and the well-vascularized regions here. At the end of this simulation, we simulated up to 12 hours. So at the end of that, the survival fraction is actually the lowest compared to the other regions. And this reflects the interaction between the drug transport, the leakage, because as I said earlier, the transport is bidirectional. So it can also, drug can also be leaked and transported back to the vasculature. So, what the conclusion here is that based on this particular analysis, that the better anti-cancer effectiveness is achieved in tumor regions with low microvascular density. Okay, so we're going to continue, and perhaps it'll be helpful for me to know whether anyone in the audience actually work in this area. I know we have some researchers, PhD students, or master students. Does anyone work on a project involves modeling drug transport or anything related to tumor? No? Okay, all right. Now, let's look at the effect of tumor size. So again, at the time that we had the opportunity to collaborate with our radiologist who who scans the cancer patient, and so he had the data for us, and these are prostate cancer of different sizes, and these were built based on MR scans of the patient. So we chose a very small tumor, and then we chose a medium-sized and a large-sized tumor. So you can see but the exact size is beyond our control because it depends on the patient. So it's just that you can try to pick different levels and so to see whether the size of the tumor has an effect on the treatment outcome. So the size here, so the small one is very, very small. And then this is the largest. And because we learned from the previous simulation that this S over V ratio, which is the reflect the microvascular density, is a key parameter in determining intracellular drug concentration and also the cell survival fraction rate. And so we need to make sure that this S over V ratio is modeled correctly. And obviously one way to do it is just to fix it for all of these. But that's not the case because, fortunately, there are experimental data exist which measured, have measurements. So these dots correspond to experimental data in the literature. Unfortunately, I forgot to cite where the experimental data came from. But if you go back to that paper, it has cited the original paper where the experimental data were reported. And the fitting occur through this data, and you can find the trend, how this S/V ratio change with tumor volume. And then using this curve, the relationship we put in, we find the S/V ratio for different cases. And so here we can look at spatial distribution. So first look at interstitial fluid property, interstitial fluid pressure. And the red, so it's all red. And this is quite typical for tumor tissues because, as I said earlier, that the tumor tissue lacks a functional lymphatic system. So the fluid, there's no effective drainage of the fluid through the system, and so that the pressure gets built up. And so pressure is very high within the tumor, regardless of the size. So you get these, if it's bigger, then it's red throughout. But then there's a very sharp pressure gradient near the edges of the tumor, and the rest of the blue is a normal tissue. So there's a big contrast in interstitial fluid pressure between the tumor and normal tissue. And if we look at the spatial mean interstitial fluid pressure, this is what we get here. So it looks like in the smallest tumor that the interstitial fluid pressure, the magnitude is slightly less than the other two. Yeah. And then we also looked at the transvascular flux per tumor volume. Now, that is quite different. So the transvascular flux is much higher in the smallest one compared to the other two. So what that means is it says rapid transvascular drug exchange in small tumors, and these may actually influence the therapeutic outcome. And then if we look at the key parameters we're interested in, these are volume averaged over the entire tumor. So looking at the doxorubicin intracellular concentration within the tumor. So the small one, again, the smallest tumor has the highest intracellular drug concentration. But if you look at the survival fraction of the tumor cells, and then that has the highest, so the lower the better. So the larger ones, the larger tumor had a slightly better outcome compared to the small tumor. So what we've learned from this analysis is that the anti-cancer effectiveness is compromised in small tumor through this model analysis. This is consistent with experimental findings. So there are experiments reported in the literature and on the effect of a treatment on different sides of the tumors. Right, so let's look at some the multiphysics aspect of modeling these thermosensitive liposome-mediated drug delivery. So I didn't elaborate these, describe this in detail before, but here is an opportunity to see exactly what this kind of nanoparticle does and how it functions. So this type of nanoparticle is known as temperature sensitive or thermosensitive liposomes. And so the way it operates is that, imagine this is a cartoon of the nanoparticle. So the particular one that we have modeled are the liposome-based. We actually, when we did the project, we collaborated with a group of chemists who actually developed the nanoparticle so they can provide some experimental data about that particular type of nanoparticle for us. So this is a kind of liposome-based temperature-sensitive nanoparticles where the drug is encapsulated inside of the nanoparticles. And then the surface is designed such that at normal temperature, it's usually fairly tight, and so that the drug that is encapsulated, loaded inside the nanoparticle, they don't leak out. But then, if the temperature is raised to a certain limit, then the surface membrane will break down, and then the contents inside will be released. So that's the temperature sensitivity of it. So how do you achieve the temperature, the heating, the required temperature? Well, this is done through HIFU, or high intensity focused ultrasound. So HIFU is actually one of those relatively commonly used technique in treatment. Not only for this particular type of treatment, but it can treat other type of diseases. I can't name them, but it is used in clinics, and the radiologist I collaborate with is actually a specialist in operating high-intensity focused ultrasound for other type of treatment. Right, so you obviously use high-intensity focused ultrasound to achieve, to get a focal region, and where it has a thermal effect. So if you focus the ultrasound, you can either produce a thermal effect, you can also have a mechanical effect. I think from the cavitation, you can create some sort of a cavitation that is also utilized in different areas. So here we're interested in its thermal effect. And then so then you can design the system so that they can focus in the region fairly precisely. So the focal region is obviously somewhere right inside the tumor. And then you only heat up the tumor, you don't heat up the normal tissues. And so once the tumor is heated to a threshold temperature, it's usually, because we're talking about living tissues, so you have to be extremely careful with the temperature. So the heating we're talking about is mild hypothermia, and typically it's controlled like 41 or 42 degrees. You can't exceed more than that. And you have to be controlled very carefully, very precisely. So imagine if it's 41, 42 degrees mild hypothermia, and then that the nanoparticle is designed to release its encapsulated drugs. So HIFU has, so there's a special HIFU machines that can, it's actually built in with a temperature controller to avoid overheating. So that is a safety aspect, which is very important. Otherwise, you burn the patient. So this gives non-invasive. So ultrasound is non-invasive. It does not harm, unlike X-rays, which obviously can damage healthy cells. So ultrasound is relatively safe. And so this is a cartoon, shows how the thermosensitive liposomes are in the system. So it carries, it can be delivered through intravenous infusion or local delivery, but it carried in the blood, and then it permeates into the tumor tissues. And then if you apply the focused ultrasound heating up to a certain temperature, and here shows around 41 degrees. So this red region is a heated area. Then these nanoparticles could be anywhere. They could be inside the capillary vessels. They could have permeated into the tumor tissue space. So then when the region is heated, then these nanoparticles are heated to that temperature, and then the drug are released. So the drug release could take place either inside or outside. So how we model this whole thing? And so first you need to have a model that simulates the HIFU heating, acoustic model. I don't think I've brought my equations over here. I did not plan to cover the particular equations that describe the HIFU heating. However, that has been, it's available. It's reported in this paper. My former PhD student did in biophysics report. So there is a whole list of all the equations that is included in the model. So that is coupled with the other equation that we mentioned earlier. For example, these drug transport model. And then in the 3 dimension as well, coupled with the transport within the interstitial fluid in the tumor tissue, and also so the fluid flow and the transport as well. But on top of that, you also have to consider heat transfer, because you heat up the local tissue, you heat at a focal point. you can't heat the entire tumor, but because of the heat transfer, because we have blood perfusion in the tissue, so that carries, that promotes convective heat transfer. So you have to model the heat transfer inside the tissue, and to get the temperature distribution. The temperature distribution will then determine the other parameters. So there is a heat transfer equation there. There is a separate equation to model the energy released through by focused ultrasound, coupled with the other equation that I mentioned earlier. Right, so this is a fundamental equation, the by heat transfer equation that you can adopt. And so there are source terms, so this term reflects the heating source. This is the absorbed ultrasound energy. And then this is the this part is the this is the heat transfer due to perfusion. So there's the perfusion rate of the blood flow within the tumor tissue. So you solve the heat transfer equations, and then if the system is built with temperature control, then that needs to be reflected in that equation as well. This is to limit the temperature to a certain limit. And then there are expressions for each of these terms. I think that the model, the equation that we reported in the paper I cited on the previous slides is more advanced, more comprehensive than what I show in these slides. But you get the feel about it. So you have these various components, you put them all together, and then you solve these. So in addition to the model that we saw before the break, the additional complexities here are not only the additional heat transfer equations, etc., but also the temperature-dependent properties. And there are a list of the properties which are dependent on the temperature, even though the temperature does not vary a lot. So we only talk about the temperature change from the body temperature to mild hypothermia, so just about 4 or 5 degrees at most. But still, it has an impact on these. The permeability has impact on the diffusivity, but perfusion rate, transmembrane rate, and also, well, if you have a temperature controller, then that makes sure that it does not overheat the body and need to be considered. And perhaps the most important parameter that influences the drug release rate, because this particular type of TSL designed to be temperature sensitive. And so this gives you an example of the release rate, how temperature dependent it is. So at 37 degrees, the release is not zero. It is it's very difficult to design a nanoparticle that does not leak anything. So this is known as, usually when you design a nanoparticle, you test the leakage of the nanoparticle, which is also related to the stability of the nanoparticle. So at the normal temperature, there's still some amount of leaking, but it's a relatively small amount, depends on how you synthesize nanoparticle. But that as you increase temperature, then this release increases. So this particular, obviously these parameters are not fixed. They are nanoparticle dependent, and the particular parameter that I used correspond to only one type of nanoparticles. So if you model these for different type of design, different nanoparticle, make sure that you get the relevant parameters and data for those. And other controllable factors in modeling TSL drug delivery, these include the heating duration, like short heating or long heating over an hour. And then, so this is, for example, we start off with a geometry like this. I think the example I show you is actually 2D model, but this can be solved in 3D as well. The formulation itself does not limit the dimension of the problem that you solve. It's just that it will take a longer time if you solve a three-dimensional problem. So it has the tumor inside of a normal tissue. There are focus region. So the focus region could be, it should be a focal point, and sometimes the heating is designed to the focal area actually move around a little bit in order to achieve a uniform temperature distribution. So, well, the key here is to ensure that the temperature within the tumor is as uniformly distributed as possible, as close as possible to the higher end of this mild hypothermia condition, so that you can fully take full advantage of the higher release rate of the drug from the nanoparticle. So, so we can have a look at the spatial distribution first. So this is a temperature for the given focal heating point. This is a single fixed heating point. And then you can see the temperature distribution inside the tumor. Obviously, it's easy to understand that the temperature is higher near the focal heating point. And then how these could change as a function of time with different heating durations. So this is in the tissue, and then this is in the blood at 10 minutes after heating. And then these are the corresponding maximum temperature and spatial mean temperatures under 10 minutes heating. So the maximum temperature, you can see that the maximum temperature within the tumor reach 42 degrees because there's a function of the temperature controller. It does not allow the temperature to exceed that amount. And then this is how the temperature is in the normal tissue. And this is the temperature of the blood in the entire domain. So the temperature is different because you have heat transfer. If you don't model heat transfer, then everything is heated the same temperature, right? So you've got to have heat transfer equations in the model as well. And this figure gives you the spatial mean temperature. And so that is the spatial mean in a tumor. So, well, they follow the same trend. It's just that this gives you the peak value, this gives you the spatial mean. Monitoring the peak value is obviously important to make sure that you don't overheat. And so the mean temperature in the tumor is much higher than the mean temperature in other compartments. And then this gives you the key parameters we're interested in, and that include the intracellular drug concentration and the cell survival fraction. So you can see that these different curves correspond to different heating mode. If you don't apply any heating, and that means, well, you only have the leakage of the drug, you don't have the benefit of increased release rate. So this is the black curve. And if you started heating over a longer period of times, then you get more drug released from the TSL nanoparticles. And then the impact on the cell survival fraction is over here, so that obviously goes with the expectation. So if you use a normal mode without heating, it's over here, and then if you use heating, then you have a benefit. But you probably say probably the difference is not that significant. But when we talk about treatment in patients, any small gain is useful, is relevant. So what we've learned from here is that TSL drug delivery hypothermia induced by HIFU is effective in achieving localized treatment. And if you prolong the heating durations, it helps with improved cell killing. But if you relate back to one of the first slides that I showed you when we did parametric sensitivity analysis, which shows that heating beyond 2 hours does not provide any gain. So this can be connected to that. And perhaps that's why when we did this study, there's no need to simulate heatings longer than 2 hours. So we kept that within. So it was informed through compartmental, relatively simple simulations. We also can look at the spatial drug concentration. So when you solve a 2D or 3D problem, that's what you can get. And for 10 minutes heating, and you can look at, so these different, well, they all look the same, have similar patterns, but they show different things. The one on the top left shows this is the free doxorubicin intracellular drug concentration. So these are the drugs already released through the nanoparticles, and you can see that they actually spread quite well inside the tumor. And there's only the peripheral regions of the tumor that did not get the drug. at this time, but this is only 10 minutes. And if you look at the spatial distribution over longer period of times, and these could spread out. And this is the doxorubicin drug concentration in the extracellular space outside of the tumor cells. And this is the intracellular drug concentration. So this is there. And you also have the doxorubicin, the bound drug to intravascular space. And perhaps what we're more interested in is the cell survival fraction. So look at the cell survival fraction, the lower the better. But if you look at the scale, of course, this is still quite high. So not many tumor cells have been killed just yet after 10 minutes. You need a much longer treatment. Typically, so there's no magic drug, right? You have to take multiple treatment over a longer period of times in order to achieve cell killing. Okay, I think that's probably what I'm going to cover in terms of the TSL drug delivery. And then we move on to something related, but a slightly different application. It's also about the use of nanoparticles, but in this case, it's not to deliver drugs for cancer treatment. And these nanoparticles, they don't carry any drugs. They're designed to enhance the efficacy of radiotherapy. So I'm not expert in radiotherapy, but this is what we usually know about radiotherapy. It uses high energy X-rays pass through the body, and so it can be absorbed by DNA and generates free radicals. So the efficacy of radiotherapy, it can be impaired in some patients, and this could it would be desirable to enhance the efficacy of radiotherapy through the use of specially designed nanoparticles, and these are called radiosensitizers. And these could be, they're different from the TSL that I talked about. In this work, we collaborated with a nanoparticle expert who designed these particular nanoparticles for the enhancement of radiotherapy. So they had a startup company. We were very hopeful at the start of the project. We were hoping that this particular type of nanoparticles will be successful, go all the way through to trial. So the intention of this project, which is performed by my PhD students, co-sponsored by the company, and it was to use our model to help with the design of the first trial protocol. However, as with all startups, and this particular nanoparticle did not fly, mostly due to COVID, because the key experiment was designed to take place during the COVID period, and that did not materialize, and then therefore, so the company was not able to secure additional funding to do further research on this. So that's a side story of this. But nevertheless, we had the opportunity to collaborate with the physicists who designed the nanoparticle, trying to understand the distribution of nanoparticles in tumor. So the aim of the project was to develop a multi-scale intratumoral nanoparticle transport model for application in realistic tumor geometries. And here what we're trying to achieve is that, and again, it's trying to achieve a uniform distribution of the nanoparticles. Now these nanoparticles, they're designed to be injected locally into the tumor. So we need to model that scenario. Well, the strategy here is there are some commonalities to what we mentioned earlier when we modeled the TSL drug carriers. But here the additional aspect are the nanoparticles. Now these nanoparticles, they usually have a surface charge and they can interact with cells. So in other words, we needed to we need to have a model that can also simulate the microscopic transport, and then so look at the cellular levels, and then we need to combine that with the macroscopic transport. So as far as the macroscopic transport is concerned, this is nanofluid transport within tumor tissue. This is actually very similar to what we covered before. So you can use like Brinkmann equation to describe the interstitial fluid flows, and then use the convection diffusion reaction equation. And the only difference is here is that these nanoparticles are not consumed. So there's no reaction here. It's just a transport. And then the other difference is that there is a needle that inject the nanoparticles into this space. So this part is actually quite similar, quite common to the model that we already described. And this part is relatively new, and then so you need to look at the cellular level, and then so the cells, look at the cell surface, and there's a thin layer around the cell surface, so there's a spherical sort of shape. And you model the particles, the nanoparticles, inside that space. And the purpose is to calculate the deposition rate of the nanoparticles. Because if we look further at the macroscopic scale, the nanoparticles, looking at the right, that this is the cell surface. Nanoparticles are much smaller in size compared to the cells. Now, tumor cells are negatively charged. These nanoparticles, they can be designed as no charge, positively charged, or negatively charged. So if these particles are negatively charged, then there is a repulsive force between this particle and the cell. And then if they're positively charged, they have attractive force. So in other words, these electrostatic double layer force need to be accounted for. And the other forces and balance of the other forces, you need to write an equation. So I've only put the simple equation here, but there is lots of terms in this F. You have to write out the forces that we need to consider at this scale. On this scale, this include drag force, Brownian force, Van der Waals force, and also lift force, electrostatic double layer forces. So all of these forces need to be expressed and then evaluated and put back into this equation, and that will give you the velocity of the particles, and then you can track the trajectory of the nanoparticles. So you need to model a large number of these particles to get a statistically converged deposition rate. It is the deposition rate that we need in order to simulate the nanoparticle transport within tumor. So there's assumptions here. The assumptions include the particles are spherical, which define these nanoparticles are actually they're spherical. But the particle-to-particle interactions are neglected. We only considered particle-cell interaction, but not particle-particle interaction. And this model is actually, we did not design this from scratch. This was based on a well-known Happle cell in sphere model. You can find this in the literature. So if you wanted to understand all the forces and how each of these the force components are actually evaluated, you can look at the papers where all the forces have been expressed out. Right, so from there, we get a particle deposition efficiency, which then goes into the transport equation, and this is where it comes. So in the transport equation, we look at the transport of the nanoparticles within the tumor tissue, and so this is treated as a source term. Imagine that if the nanoparticle is deposited on the cell surface, then it's no longer available for further transport, right? So it needs to be taken out. And so that's why it's here. And then so in this expression that the connection efficiency, so this is I think this is a connection efficiency, yeah. So this one was evaluated from the microscopic particle tracking model that I described earlier. Right, so then you can put this all together. The way we usually do is you run the microscopic particle tracking first. You run it for different fluid velocities. You run it for different nanoparticles, and then you're trying to get the trend. I think I'll have one later on. I'll show you in a minute. So we built a tumor model. This is a realistic tumor model. This was reconstructed from micro-CT images obtained for the corresponding experiment. This was a lab-grown tumor. And This is the longest axial distance is less than 16 millimeter. And they used a bevel tip needle. So we modeled the actual needle like this with the exact dimensions and then put inside. So obviously with this type of design, and you can imagine that the tumor can come in any shape or form. And so there are a few things you can play with, like the shape of the needle, the type and the size of the needle. And also perhaps most important is the depth of the needle, so where your injection point is. This injection location is probably quite important. It's going to affect how uniform or non-uniform the nanoparticles are going to be distributed in the system. So you can play with, this is one of the parameters that you can optimize with in silico simulations. You can do many simulations with different injection locations, and one idea that we discussed with our collaborator at the time was also to change the location around during the injection. Right, so, and you need to feed in some appropriate boundary conditions for the nanofluid convections and also for the nanoparticle transport. So you start with some parameters and then you run your simulations, and these are some of the results. And I'll show you some of the key parameters needed to run the simulations. You need the particle diameter. This was the actual experimental particle diameter that we use at 60 nanometer. And the particle diffusivity is a very important parameter, and it was found that if you use uncharged, zero surface charge nanoparticles, then its diffusivity is actually several orders of magnitude smaller than if you use charged particles. So obviously the charged particles will transport better in this system. Now, the results of running the microscopic particle tracking model look like this. So what you get here, I don't know whether you may not be able to see it clearly. So you see the collection efficiency, which is a parameter that we need to include in the transport model. The collection efficiency as a function of the fluid velocity. So this is for negatively charged nanoparticle. This is zero surface charge. This is for positively charged nanoparticle. So now, we have several symbols here. The one that was obtained from the simulation, these are the dark, this one's diamond. These are the simulations. And you also see a few other symbols labeled as TENG or some others. And the label I explained over here, because in the literature you can find some correlations that allows you to estimate the collection efficiency based on existing theory. Well, we obviously, in the early days, we used their correlations, but we were not too sure how accurate the correlation could be, so we ran our microscopic particle tracking. But you can see that actually the correlation gives you very different results. So it turns out that it is necessary to run the microscopic particle tracking model in order to get more relevant and more accurate predictions of the collection efficiency. Now, if you compare these, one other thing I wanted to mention is that if you look at the zero surface charge nanoparticles, and you will find that the simulation results actually agree with some of the correlation, and that's because the correlation will probably derive for zero surface charge nanoparticles. When you consider either positive or negatively charged nanoparticles, then you have to run the simulation. And for positively charged nanoparticles, there's no correlation, so these were based on simulation results. I think what we observed from this simulation is that the collection coefficient decreases with the fluid velocity, so that this trend is the same for all these particles. And then the other thing is the particle surface charge has a significant influence on the collection efficiency. Now, if you just look at the trend, they seem to be the same, but the magnitude is actually very different. If you look at the values, the positive charged nanoparticle, this scale is 0.45, and then and this is for the no charge the particle, and the scale goes up to 0.4. Whereas for negatively charged nanoparticles, this scale is actually very small. So what it says is that in terms of the magnitude of collection coefficient, negatively charged nanoparticles has the smallest, the lowest deposition rate, which is obviously desirable in this. Well, whether it's desirable or not, you have to see it from two sides. But from the transport point of view, you probably wanted to have a low deposition rate so that the nanoparticle can spread more throughout the tumor rather than get concentrated within the injection point. So just a few examples to show the spatial distribution. Sorry about the text is probably a bit too small to read. So what we see here is that so we see the distributions within, so the top row, I think they correspond to the concentration within the fluid, and the second row, the deposited particle concentration. So if we look at the deposited particle concentration first, these are the contours, and these correspond to a time of, well, they actually correspond to slightly different times. For the negatively charged nanoparticles, these are captured at time equals 10 seconds. For zero surface charge, these are captured after one. Sorry, the time scale is actually different. You have to read the times for each one of those. I think my student chose at different time points so that only at that time point that you can actually see a distribution, otherwise it's probably the same color. So if we look at the pattern, the deposition pattern, obviously for positively charged nanoparticles, a lot of the particles are actually deposited on the surface. They stick to the surface, to the cell surface, around the area of injection. Whereas this doesn't happen to the negatively charged nanoparticle because there is this repulsive force, and so that nanoparticle can spread a bit more inside. And then if you look at the nanoparticle distribution within the fluid, there is obviously a very, this is very different, and this kind of tells you the availability and the spreadability of the nanoparticle within the fluid. So negative charged particles performs better than the positively charged or no surface charged nanoparticles. I think this just summarized what I said, that the negatively charged particles spread much further, and but if you look at the local distribution with this particular type of a needle design, that behind the needle, that because the shape, it blocked the flow to the back of the needle, and so that you have less concentration behind. So that's something maybe through design a better tip, this problem could be improved or resolved. Right. So with the model, you can do experiment with a lot of the things. As I said, you can try different injection rate. For example, increasing the rate will improve the local convection, and to what extent that will improve the spread, and it's something that can study as well. I think I have one more slide on that, and this is about what if you change the injection location. So the one that in the previous slides, that the injection location is somewhere over here, but then if you say, well, this is the longest axis of this tumor, and then you can't change the needle. Well, I think the needle depth is probably fixed, but then if you move it towards the halfway towards the middle, then what distribution pattern will you get? So these are the distribution patterns corresponding to negatively charged, zero charged, and positive charged nanoparticles. But then the problem you can realize immediately is that by moving this injection point to the middle, then it becomes too close to this edge, and so that you lose some of the nanoparticles to the so they're leaked out into the surrounding tissues rather than get retained within. So obviously the point needed to be considered very carefully, and they could be optimized to a different shape of a tumor. And you can also do some more quantitative sort of analysis by looking at the total volume of deposited particle volume of the particles relative to the tumor size. And so that gives you a clearer sort of comparison about the difference of the injection location. So one of these corresponds to the end, which was on the previous slide, and the middle is this one. So this the middle is the middle way along this longest axis, and you can see that changing the location actually can change the percentage volume of the particle deposited quite significantly. So there is a lot of room for this to be further optimized. Right, so for the macroscopic particle transport, I think that's all I wanted to say today, and I'll just give you a few slides to give you a little bit of flavor about system-based model for drug transport and cellular uptake. Now, first I want to declare that this work was done in collaboration with my colleague who was in the same department, Dr. Krishnan. So originally he's Indian, and then he studied his PhD in Princeton, and then became an academic professor in our department. So he's a specialist in system biology. So any model, anything that I talk about in the next few slides, if they're related to system biology, I would attribute that to him. So I collaborated with Dr. Krishnan in this project. We co-supervised PhD students at the time. So that he contributed to the system biology modeling aspect, and I contributed to the transport modeling aspect. So together it worked really well, and we published quite a few papers in this field. So where does the system biology comes in? If we consider, so here this diagram probably look familiar because it's kind of, in terms of the concept, is very similar to the Croft cylinder model when we look at the oxygen transport. So here this is in the cancer drug delivery field, this is known as a tumor cord model. So you consider a tumor tissue, a cylindrical tumor tissue surrounding a blood vessel. So this is a capillary vessel, and this is tumor tissue. And then the drug is carried through the vessel, and then it can transport across the vessel wall, enter into the tumor space. So we covered the drug transport in the previous slides, and this is still the same. In terms of the drug transport through the vasculature within the tumor tissue, still governed by the same equation, and that gives us, solving these equations give us the spatial drug concentration distribution and also temporal distribution as well. So that's fine. And however, the way that the effect of the drug on tumor cells, I remember that previously we simplified that using an equation, using a pharmacodynamics equation, where the tumor cell density is expressed as a function of something related to intracellular drug concentration. And that is obviously tumor cell dependent and drug dependent. So, but if we understand enough about this cellular signaling pathway, then this can be programmed into the computer model. And this, what it does is that it can predict the fate of the cells. So whether the cell will be killed and whether the cell would survive, and then find a way to mathematically feed this information into tumor cell density. Right? So in the previous model, we're missing this part. So basically this part has been replaced by this equation based on experiment or based on empirical sort of relations. By including system biology, then this can be more precisely determined. And then that's fed back into the macroscopic sort of model. So the advantage of using model the system level is that it can provide mechanistic insight into drug effects on tumor cells. And it can also be used to investigate multiple drug resistance mechanisms and how they interact with different drug stimuli. So I'll give you a few examples, but without going too much into the system biology side. The model is still very familiar. Tumor cot model with a vessel inside, with tumor tissue outside. So initially, so this is the when you decide the modeling strategy, and so you have to make up your mind about what the focus, what questions that you try to address. And here we wanted to see how the system biology cell signaling would impact the cell survival and in the prediction. So we have to sacrifice, because you're introducing additional complexities, you have to sacrifice the model complexity in other aspects. So we went back to a simple geometry. We're not going to do a three-dimensional realistic tumors because then it becomes mathematically very difficult to solve. So come back to a simple geometry, but focusing on the cellular signaling side. And then if you look at these schematic diagrams, so only consider diffusion, the top, and then the consumption by the tumor cells, and this is determined through this bit. So you still need to have the intracellular drug concentration, but then include the signal transduction, and then that predict the cellular response, and then in terms of the cell apoptosis. And then eventually it fed back into reduction in the tumor cell density. So this paper that we jointly published with Dr. Krishnan as well, with our students, has all the details about the system biology side of the equation that we introduced. So it was a relatively basic model in that respect. So we introduced a coarse-grained description of the apoptosis signaling, and we used the threshold effect and irreversibility in the model. And I'm not going to talk about the details of these because that's Dr. Krishnan's work, and I just want to say that by modeling these, it gives you predicted the cell response. So the R is the response. And this cell response is then reflected in eventually this equation that describes the tumor cell density. So how the tumor cell density change as a function of time, because this is what we wanted to monitor. Because ideally, when you have treatment, you want the tumor cell density to decrease as a function of time, right? So this will give you a prediction of how these tumor cell density will change as a function of time. And so you start off with an initial distribution of tumor cells, and then you run your model and you see how these tumor cell density change when considering the relevant cellular signaling pathways. So as an example of application, we modeled two different types of resistance, so drug resistance mechanisms. And one is the standard negative feedback resistance mechanisms on the right. So basically what it does, it says that the drug is actually digested within the cells without killing the cells. So that could impair the efficacy, the treatment efficacy. And this is mathematically can be modeled in this way. So there's internal digestion of the drug. And this type of ABC transporter mediated resistance, it functions differently, because what is known about this type of resistance is that instead of internally digesting the drug, it actually pushes it out. So when the drug finds its way inside the tumor cells, and this resistance, this is an acquired resistance, it does not exist in the first place, but after a certain duration of the drug use, that this resistance can be built up. And then in the presence of this resistance, the drug is pushed out. So it's like a little pump. I pump the drug out of the cells so there's no consumption here. So these can be modeled and to see, and coupled with the transport, to see how these can affect the efficacy at different levels. So first we look at the behavior at the cellular level. So all you need to look at are so compare the figures on the left to the figures on the right. And if you just focus on the red curves, because the red curves model the two different types of drug resistance, and these are the responses at the cellular level. So if we look at the right one first, so this is the conventional negative feedback resistance mechanisms. If you look at the cell density starting from the bottom, now the cell density, this is normalized, and cell density remains constant at 1. What that means is that none of the cancer cells is killed in this case, and well, that's probably expected because of the resistance here. And if you look at this one, this is the intracellular drug concentration. So inside the cells, so initially this concentration increase. But because of these resistance mechanisms, that it internally consumes and digests some of the drugs so that the concentration would drop. And then let's look at the concentration of the drug in the extracellular space. Now, because you don't have, so this basically just follow the same pattern. And there is a reduction in the drug concentration in the extracellular space because of the consumption of the cells by these resistance mechanisms. Look at the other type of resistance mechanisms. Now, in this case, at the cellular level, that the expression effect is the same. So there is no cell killing. The cell still survived. If you look at the intracellular concentration, it's the same. It increases, and then it starts to decrease. And the reason for this decrease is because these resistance mechanisms pushes the drug out of the cells, so they get pumped out. Now, because the drug is pumped out, then the extracellular space actually collects these drugs. So if you look at the extracellular space, and there is increase, and it can sustain this level of concentration. So this curve is quite different from these mechanisms. And then the next question is, does this difference in the cellular level actually, how does that affect at the tissue level? Now we look at the tissue level using the tumor cult model. And then I think if we look at the one at the top, so this is the, so A is a snapshot of spatial extracellular drug concentration distribution. And all of these curves correspond to the ABC transporter-mediated drug resistance. So if we start from, now, this is the distance. This is a normalized radial distance from the capillary wall. So in other words, the closer to the capillary wall, because here the drug is transported from the capillary and then transported along the axial direction, along the radial direction to the tumor, through the tumor tissue. So this is the far end of this tumor cult model, and this is a close end. Ideally, we would like to have a uniform distribution, and also to make sure that the drug reaches the far end and not just the close end. And the interesting thing is, because of the pumping out mechanisms of this drug resistance, it actually helps to ensure a more or less uniform sort of distribution of the drug throughout the tumor tissues in this direction. And if you look at panel B, I think that is for different times. So these are the normalized intracellular drug concentration at different times. And then if you look at 1 hour, this is the blue line. So initially there is a reduction of these drug concentration along the distance from the capillary wall. But then these trends actually change over time, and that is because some of these, the later times, it follows the pattern of the extracellular drug concentration. I think the most interesting thing is actually this panel, the bottom right, and these look at the normalized tumor cell density. If we look at the normalized tumor density, and this S represents different strengths of the stimulus. So strengths, I think in system biology, it can be some of these concepts is rather abstract, but you can kind of relate these to the dosage of the drug is correlated. And so that if you have a strong signal, so if you increase if you start with the baseline strength, that is the blue curve, so this one, there is no cell killing. So if there is not enough stimulus, then it stays like that. So that the effect of this drug resistance is to prevent the cancer cells from to prevent the cancer cell death. However, if you increase this signal by a certain extent, and you can find that this red curve, it actually, even though that there's no cell death close to the capillary wall, but there is a cell death away from the capillary wall. Now, this mechanism is due to the availability of the drug being pumped out and spread within the extracellular space, and so that so to have this effect. And then, if you increase the signal further, then you can have cell killing from the capillary wall, the near end to the far end of the capillary wall. So it's very interesting because this one helps to understand the mechanisms of this drug resistance, and perhaps help to design how to overcome this particular type of drug resistance by changing dosage and by changing other protocols that we can control. And you can numerically simulate this. And this cartoon actually explains the two different mechanisms, and one is the internal consumption, and so the effect is limited to within close to the vessel wall, whereas here the pumping action, so this is the pumping action, actually made sure that the drug is actually pumped back into the extracellular space, and then again be transported, and then to a certain extent it can help the drug to be transported further along the tumor tissues. Right, and so that was the basic model, which does not account for convection. So later on, that we extended the model to include the convective effect, and that obviously involves a lot of these equations for different regions, but still for these very simple tumor cord geometry, but just to account for the convective effect. And all of these has been reported in our previous work. So I think that's probably all about, before we finish, I wanted to show these because you should be able to understand these quite well, having done the first lecture about the mass transfer. So I mentioned about an important dimension is parameters, the Pickley number. So in this case, using the model, we can also determine the Pickley number, and that gives you some flavor about the mass transfer mechanisms in this case. You can see that the Pickley number close to the capillary wall, this is 0.8, close to 1. And what that means is both convection and diffusion are very important at this point. And then as you move along away from the capillary wall, that the convective effect is reduced and diffusion becomes, the convective effect is reduced. So that shows. But still, within this range, both convection and diffusion need to be accounted for. Right. Okay, so this is a brief summary of what I introduced you today. Looked at the PK/PD-based compartmental models. These are mathematically quite easy to solve, and they're very useful to have some sort of qualitative understanding about the interplays between different transport and kinetic steps, and identify the controlling parameters. And then we look at the continuum-based models. Continuum-based models are computationally more demanding, but they offer spatial profiles and look at the distributions of the drug, and also can account for heterogeneous effects as well if you have that information. We looked at system-based models just superficially, and then it can help provide some mechanistic insight into the drug effect on tumor cells. Obviously, a multi-scale model is needed, so that need to couple everything together. I think this is probably the time for that, because the computational techniques advanced, the numerical algorithm has advanced. So it is feasible now to try and to incorporate all these different components into the same model. But what we have not touched on is we have not considered the very complex micro environment in the tumor, and also the different type of tumors. So different type of tumors will have different response to drugs. So that also need to be modeled. And before we go, I would recommend you to, if you're interested in mathematical modeling of drug transport, I would recommend you to read this review article. This is fairly recent. It was published last year. They may have updated the review, a new paper. So this was authored by, this is my, this name was seen frequently in my slides. He was my former PhD student. So he did the project as his PhD thesis. He finished PhD about 10 years ago, so he's an independent academic faculty now in the UK. And then this is my other PhD student, and also so she also moved and as established independent researcher now. RJ, he's in IIT. I met him a few times. I examined his PhD thesis. So he has a lot of knowledge, expertise, and he continues to work in this field. And he started collaborating with my former students. So he visited the UK a couple of years ago. So he's based in which IIT? This is, yeah, right. You can find if you have an interest that I would encourage you to get in touch with him. And then so that you get involved. So this involves a lot of collaboration, and this is also a very active, a very talented young researcher in the field. He's published some highly influencing papers in drug delivery of cancer treatment. Okay, so with that I'll finish. Thank you very much. If you have any questions.
Intracellular drug concentration.
In what?
Intracellular drug concentration.
Drug concentration.
Yeah, drug concentration. Is it, can it be significant considering the effect of vibration? Like vibration in a sense, giving external equipment to vibrate our body or something.
Vibration in what you mean in cancer treatment?
In peak drug concentration, intracellular drug concentration.
The peak drug concentration.
Yeah.
Yeah. So what sort of vibration you're talking about? Are you talking about physical?
Yeah,
or
physical
numerical?
like external vibrational equipment attached to your body or at the tumor location.
At the tumor location. How? Sorry, I need to understand your question. So this sort of vibration is you mean placed in the external.
Maybe in the drug delivery itself, if we have oscillatory motion, if you provide that oscillatory motion, will it be useful in that case or not?
So it's internal oscillation for the transport or physical sort of.
The drug is being delivered.
Yes, that's right. Yes. So the drug is delivery is so we consider most of the times is intravenous systemic delivery. So it's like intravenous injection. Yeah.
If we provide some sort of oscillatory motion.
Oh, through the use of the acoustic field or some other things. That's what you meant, or magnetic field.
Basically, that is better with whether it is mixing with it or not. Like.
Oh, I see what you mean. Yeah. No, that's an interesting concept. I've not seen that being.
Because like that kind of at many places in mechanical applications or civil applications that we see, we want to have a little chaotic vibration, like concrete mixer, where we want to bind this sand, cement, and concrete.
Yeah.
So we have to provide that kind of motion so that it will be uniform mixture.
Yeah.
So that sort of thing have a better.
Yeah, I think I remember. I think that there might be it could be relevant. I think there's some work about drug delivery to brain tumor, and especially in that space, there's difficulty to overcome, for the drug to overcome the blood-brain barrier, and so acoustic techniques being used. I think the purpose is to try to promote some local movement, and maybe the cavitation effect as well, to break the blood-brain barrier so that the drug can penetrate into the area that otherwise it could not reach. So that's, I think there is a space for application. So, but it's mostly in brain tumor. Yeah. Can that be achieved through ultrasound, focused ultrasound, right? It's probably not the high intensity, maybe low intensity to create that sort of vibration. Because high intensity has a thermal effect. And if you apply that on the brain, I guess that can be a dangerous area. Yeah.
Any other questions?
In previous lectures, you said that geometry is essential for
modeling these things. But in this case, where we are talking about tumors, and in the tumors, the vasculature is definitely very thinner or small in diameter compared to the arteries case that we discussed in thrombosis,
Yeah.
etc.
Yeah.
So in your MR scans, you showed us the 2D models that you got. But up to how extent that could be helpful for designing models that will tell us how these drugs or these things are changing the or affecting the tumors precisely.
Yeah, I know what you mean. Yeah, so just model the tumor itself, the tumor tissue, without model explicitly the vasculature. Obviously, it's a very crude assumption, and that's why later on I introduced a model where you can divide the tumor into different zones based on knowledge of. It was, again, that's based on images, so that you can have a heterogeneous vasculature distribution. So in other words, in a model that on a macroscopic scale, that the exact vasculature was not included, so they are lumped together and modeled through leakage, through vasculature density, which appears as a source term in the momentum equation. If you see the momentum equation, you have a source term. The transport equation, you have a source term, and then so that it appears there. Coming back to your question, the vasculature, yes, the exact shape of the vasculature, to what extent that would affect the drug transport, I think it's difficult to tell. But I'm sorry. Inside, I know, inside the models, yeah, because there are a lot of those. And unlike the large vessels, because if you use the imaging technique, you can see the large vessels quite well. And just to give you some idea about the spatial resolution, if you use MR, the spatial resolution, the smallest you can get is probably 1 millimeter. But then inside the tumor, that the small vessels, we're talking about capillaries, so they're very, very small. They cannot be seen, so the vessel cannot simply be seen by MR. However, there's different group of different category of model where the angiogenesis is actually modeled. So the vessels, so the angiogenesis is you have a mathematical model that actually grow vessels inside the tumor, and then you have different control functions which allow you to grow a normal vasculature or abnormal vasculature. And then you can train the model to grow a vessel that represents the vasculature inside the tumor. So that's a complete different type of models. I didn't mention it here, but one of my PhD students actually did a PhD thesis on that, but it's too much to pack into one lecture. Yeah, that was a few years back.
And in the HIFU concept, what is the cutoff that we can model? The region where we can focus the ultrasound in terms of size or volume?
The high intensity focused ultrasound.
Yes.
Yeah. What is the focus of the region?
What could be the cutoff focus in terms of size or volume that we could focus this ultrasound in particular?
I don't really know. So if you say in relation to actual treatment, I don't really know. So
in terms of modeling.
In the model,
yes.
Oh, in the model there is no cutoff value. So because these are the macroscopic models, then you can build a small tumor model, you can build a large tumor model. It doesn't really matter. If you have a large tumor model, you probably need a large mesh, just like if you do any CFD work, if you have a complex geometry, then you probably need more mesh. And here you just need to increase the mesh, and then it takes more time to compute. But I don't think there is a limit as such. But having said that, I think your question probably comes from the angle of the HIFU give you a very focal sort of heating, right? And if you have a very large tumor, then you may not be able to heat up the entire tumor. Is that where the question came from?
Vice versa too, because if you have very small tumor, I don't know the limitations, but is that possible to focus to that?
Yeah, you can. So the focal point, as far as I know, I'm not a HIFU expert, but as far as I know, the focal point can be very small. Yeah, so you don't focus the large region. It's much smaller compared to any tumor size that you can spot on image. So that's fine. But then you want to. So what is important here is you want to limit the region of heating, right? So if you use it to heat a small tumor, then you control the intensity of heating. So high intensity focused ultrasound, there are a few things you can control. If you control the acoustic sort of intensity, then for the small tumor, and that will help limit the region of heating. And if for the larger tumor, you probably need to apply a higher intensity. So I think that's something that can be controlled.
Follow-up question to that. In your diagrammatic representations, the focal region where we are concentrating, the geometry of that is always either oval or circular. But in most of the biological conditions, the tumor shapes will not be in that symmetry or
exactly.
So how do we account for that?
Well, that's where the computational model could help. And I was at the time I was thinking about optimizing the heating, so the heating that you're not limited to one specific spot. You can design a system where you can move around. So it depends on the shape of the tumor. If you could design the injection point or a trajectory of the focal point, and to ensure that the tumor is heated up as uniformly as possible. That could be, I think numerically you can actually do that experiment. Yeah, I don't think it has been done yet, but I can imagine it's not something impossible. It can be done. Yeah.
Thank you.
Mm.
Any other questions? If there are no questions, I thank Professor Yun for giving this interesting lecture. So we'll again meet tomorrow at 10 o'clock. Thank you all.